creatures looked from pig to man, and from man to pig, and from pig to man again, but already it was impossible to say which was which. George Orwell, Animal Farm. In Orwell's cautionary tale, we witness a hopeful revolution spiral downward at the hoof of a corrupt and oppressive regime. As a tribute to the tragically optimistic creatures of Animal Farm, and to the lovely humans still fighting real authoritarian regimes, for this experiment, I will extract DNA from a pig's heart and also show the effect of the biological enzyme catalase on hydrogen peroxide. Let's start with the DNA extraction. Now, when we cut the pig's heart in half, this will be a physical change. Pig's heart is solid as a state of matter, and so the particles composing the heart are tightly packed together. They remain in a fixed position. There is catalase half. To prepare the heart for the extraction solution, I'll cut this into cubes and then puree the cubes in a food processor until a cohesive paste is formed. So to prepare the extraction solution, combine the sodium chloride, the pineapple juice, the dishwashing soap, all into the distilled water. Water is a liquid, and the particles composing a liquid are close together, but they move freely and they take the shape of the container that they're in. Now we're mixing in 3 solutes to this solvent water that will create a homogeneous mixture. So these particles will distribute evenly throughout the mixture. And so after this is mixed thoroughly, we will pour part of our extraction solution into the bag with pureed pig heart. So when that is dissolved, the nuclear contents, or the DNA, will be released. Now, from this homogeneous mixture that we had right here, we've added in this pig puree, this solid matter. This is not going to dissolve seamlessly into this mixture, so now we've created a heterogeneous mixture. So we will continue to mush this around for several minutes. So in the mixture that we created, the purpose of the sodium chloride is to neutralize the charge of the DNA molecule, which will help the DNA become less water soluble. And the pineapple juice breaks down proteins that are found in the cell that could damage the DNA after it's released from the nucleus. And the jury is still out on whether or not the interaction between the dishwashing soap and the cell membrane is a chemical change or a physical one, while the phospholipid bilayer is really just rearranging. The release of the cellular contents could result in chemical reactions unknown to me. Right now, we are creating another physical change. The liquid is being filtered out of the solid. The chemical makeup of this mixture remains the same. The physical state of it has changed because now we are left with solid matter and the liquid that was contained within it. We'll pour our straight liquid into a test tube. And now we will take ice cold isopropyl alcohol and float it. You can already see the strands, white gloves, the strands that are forming over the surface or within the clear isopropyl alcohol that are floating at the top are DNA that has precipitated. This gross blob that you see between our solution and the isopropyl alcohol that's floating right here is our solid DNA precipitate. It once was microscopic inside of all little Mr. Pig cells, and now we can see it unwound at a macroscopic level. Catalase is a biological enzyme that breaks down hydrogen peroxide that is produced by the cells in the body and turns it into oxygen and water. It does that in the human body. It also does that in the pig's body. So we have here blood and pureed heart from our pig heart from earlier. And we also have some hydrogen peroxide. So we will mix the two together. So we've got liquid hydrogen peroxide, liquid blood, solid heart puree, and also I should note this is 12 volume, or I'm sorry, 40 volume, so 12% hydrogen peroxide. It also contains phosphoric acid, so I feel like I'm taking a little bit of a gamble here, which I know is against the rules. If I explode, it's been a pleasure, and I've not known you long enough, so thanks for the class. But no explosions, and you can see that the catalase reacted pretty aggressively and pretty quickly on the hydrogen peroxide, producing little bubbles, lots of foam. So that is oxygen gas and water, and it looks really cool. So now we have blood, heart foam. It is Final note on our pig foam, I think I failed to mention that this is a chemical reaction between enzyme catalase and our hydrogen peroxide. The